Hello from my side. My name is Nikolaus Fortelny and I'm from the Research Center for Molecular Medicine in Vienna, Austria. I will be talking today about how we build neural networks that mimic biological networks in an approach that we call knowledge-primed neural networks. The idea behind this approach is that from multi-omics and single-cell experiments we can generate very large quantities of gene expression profiles for cells, and also their phenotypes. For example, are cells stimulated or not, or different cell types. Now, we can use machine learning to connect these gene expression to phenotype, but the functions that we use are generally not much to do with biology. Especially neural networks have these different layers of neurons and nodes that are not easily interpretable, and once we have trained weights, it's hard to tell how we can interpret them, and this is why neural networks especially are generally considered a black box. Now, we thought that we do know a lot about these functions that connect gene expression to phenotype, especially there are regulatory networks of signaling proteins and transcription factors that regulate gene expression. And so we build networks where the neural networks are mimicking biological networks, we train them, and then we extract weights from them. And what we then want to do is to identify the nodes in between, and nodes correspond to proteins. And so we identify the proteins in between that correspond to regulatory proteins that are relevant for a biological process. And this is, of course, a very important task because it's very hard to identify these regulatory proteins, the mechanisms that link phenotype to gene expression. To identify these proteins, we calculate node weights. So these node weights quantify the importance for each hidden node, so in the hidden layers, for the prediction. So they connect the predictive features to the output, and thereby they are proteins that connect the phenotype to the gene expression. To show that this works, we generate a very simple simulation where we have just one important node, and when we repeatedly train this network after random initiation, we see that the node weight for this particular node is always higher than these other nodes, which are not as predictive. And now I would like to discuss two adaptations to the general deep learning or neural network algorithm that really enable this kind of interpretation. The first one is in the case of redundancy. So to show what we do here is we generated a simulated network where there are two important hidden nodes, A and B, and we show that if we use general training, then both nodes generally get higher node weights, but those are inversely correlated if we train the network multiple times. What this means is that some networks will put a lot of focus on B or on A. To alleviate this, because this shouldn't be, because those two nodes are by design designed to be equally important. So to solve this problem, we employed node dropout. So dropout is a common algorithm in machine learning or in deep learning, where nodes are randomly dropped during training. And so what this forces the network to do is to spread weights across different nodes, and so it increases the correlation of node weights over multiple random initiations, and it forces those to be highly correlated. The second adaptation we used is to control for network connectivity. Because biological networks have a very uneven connectivity, there can be some proteins that are connected to many other proteins and some to very few. This can have impacts on these node weights without any data, so just the network structures. To show this, we generated control inputs where all genes, or all input features, have the same level of predictiveness. And so then when we train networks on these data, then the node weights just correspond to the importance that's just due to network structure and not to the input data. When we then compare this to a network constructed on a realistic data where there are some predictive genes but most are not, and then get the node weights from the real data, and when we compare those two node weights, then we can see what is really due to the data and what node weights are just due to the network structure alone. Now, with these two adaptations, we wanted to see how this approach performs on real data. And so we chose a data set on T-cell receptor signaling, which is a great data set because there's a lot known about T-cell receptor signaling. So we had T cells that are stimulated and unstimulated, and we profiled their gene expression, and then we predict stimulated from unstimulated cells using our approach with a neural network that mimics a biological network. So what you see here is the full network. And what our approach showed us is first that we can accurately predict stimulation from gene expression in this network. And second, it highlighted a few of these intermediate proteins as very important in our approach of dropout and control inputs. These were highly enriched for known regulators of T-cell receptor signaling, and this enrichment was gone after we shuffled this network too. So this showed that we can use those predictions, first because predictions are accurate and because they are gone after shuffling. Also, we observed that these regulators are not observed in the transcriptome, so what we identify really are novel regulators on a protein level and not just changes in gene expression. So we really identify this protein level, which is important. Now, since then, we have tried to extend this approach and applied it to more complex data sets. So what I'm showing here is one data set from the human cell atlas. So this is a large data set with 500,000 transcriptomes, 3 different cell types and 2 different organs. And again, here we were accurately able to predict cell type from gene expression, which is actually quite an easy task, as many people have shown. But we were able to then interpret our network and identify the important proteins, and also these results were again interesting and validated our approach. In particular, in the bone marrow, we observed a lot of nodes, proteins that had to do with developmental biology, pluripotency, and proliferation, which is what happens in the bone marrow. While in the cord blood, we observed more differentiated cells with specific biological processes, which is also what we expect. Now, with this, I have shown that we can use high throughput data, especially single cell data, to predict phenotype from gene expression. And if we do this in our approach of knowledge-primed neural networks, we can then interpret these networks for regulatory proteins. And I've shown that this approach requires repeated training with dropout and control inputs to really yield meaningful results. With this, I would like to thank my PI, Christoph Bock, and the lab, as well as other people in Vienna, and EMBO for funding. And I would like to point out that I will be starting a new group in Salzburg as assistant professor, and I will be looking for postdocs, PhD students, and staff scientists. So if you're interested in this kind of research, please write an email. Thank you.